I'm one of the editors. I'm one of the editors on the Journal of Experimental Botany, and it's a great pleasure this afternoon to invite Ying Li from Purdue University in Indiana to present her paper that was published on the 4th of May edition this year. And she's going to be talking about epigenetic integration of primary and secondary metabolism to channel volatile production in petunia flowers. And without further ado, I'll hand over to you.
Thank you, Rob, for the introduction. Can everyone hear me okay? Okay. Yeah, I would like to thank the organizer, Society for Experimental Biology, for this opportunity to talk to you face-to-face virtually about my recent publication. So that is about the dynamic chromatin modification in petunia flowers that regulate temporal fluorescence emission. So when we smell the flower, what we actually perceived is floral volatile organic compounds, short as VOC. So flower emit a lot of those compounds from the petal, not to make us happy, but to attract pollinators. So this is a question with a lot of relevance, with reproduction success, which is important for agriculture as well as for ecosystem. Using petunia flower as a model system, the last 20 years of work has generated a pretty detailed image of the metabolic network that is underlying this floral VOC biosynthesis. And that include the metabolic pathway from the primary metabolism, which mostly concern the biosynthesis of phenylalanine, the aromatic amino acid, that is used as a precursor by the secondary metabolic pathway, which is the VOC biosynthetic pathway, to convert to a variety of different volatile compounds, which are highlighted in red here. So when we say we smell the flower, we actually smell a unique mixture of different metabolites, and that is a signature scent to attract a specific kind of pollinators. And as you can see, this VOC biosynthetic network is rather complicated with multiple branches driven by many different enzymes. And in collaboration with the co-author of this paper, Natalia Dudareva, also here at Purdue, we are interested in the metabolic profile that is controlled by developmental signals. So using petunia flower as the model system, when we follow the developmental process of the flower, on day 0 of emphasis, the flower is closed. We call this bud stage. And when you measure the emitted metabolite at this day, no matter when you look at the precursor phenylalanine, all the emitted VOC show with one representative VOC compound here, phenylacetaldehyde, which is here, their level is relatively low on day 0. And 2 days after that, the flower open and it's ready for pollination now. And we can see that the metabolite level increased greatly from day 0 to day 2. And this makes sense for the flower because the synthesis of the secondary metabolites are very expensive. It consumes carbon skeleton as well as consuming energy from the primary metabolism. So the flower only wants to make this compound when it's absolutely ready for pollination. So underlying this temporal profile of metabolite emission is a coordinated gene activation for genes that are function in the primary metabolism network for the synthesis of phenylalanine, as well as for genes encoding enzymes that function in the secondary metabolic part for the synthesis of the VOC molecules. When you look at the expression level of those genes, again, they are really low on day 0, and then they are co-activated from day 1 to day 2, and they usually reach their peak expression level at day 2. So our primary interest is what is the regulatory mechanism underlying this synchronized activation of genes from different metabolic pathways in the petunia flower so that the flower can make the scent at the right time of the developmental stage and at right time of the day. So when we talk about gene expression, one mechanism to regulate gene expression is chromatin modification and specifically histone modification. So a little bit into that background for people who don't think about it every day. In eukaryotes, the genomic DNA exists in the form of chromatin, and the basic unit of chromatin is nucleosome, which is 147 base pair of genomic DNA wrapped around a histone core. So here we can see a histone core has 4 different subunits, and the N-terminal tail of each histone subunit kind of stick out from the core. And the different amino acids on this histone tail can be modified by different groups, and that's called histone modification. So perhaps the most well-known histone modification is histone acetylation. For example, H3K9 acetylation means that the nice lysine on the tail of the histone subunit 3 can be acetylated. And similarly, we have H3K27 acetylation, as well as acetylation at various different substrate amino acids. And across the board, histone acetylation are generally associated with active gene expression. And histone methylation is another well-studied histone modification. And we have K4 and K36. Trimethylation, those are usually associated with gene activation. And we also have K9 and K27 methylation. Those are usually associated with gene silencing or heterochromatin formation. So for histone methylation, there can be different roles to affect gene expression. In addition to that, we have a few other forms of histone modification. And together between those different histone modification at different amino acid and sometimes with different level of modification, for example, monomethylation, dimethylation, trimethylation, you can imagine they can form a very complex histone code. And this histone code can affect the chromatin states, hence affect the gene expression level. So the histone modification is dynamic because it's being added to a specific gene location by a group of writer protein. And when it's no longer needed, it can be erased by another group of protein called erasers. So for each of these different histone modification, there is a specific groups of writer proteins and erasers proteins. Just give you an example for histone acetylation. The writer proteins are a group of enzymes called histone acetyltransferase, short as HAT. In plants, there are 3 gene families encoding histone acetyltransferase. GNAT means CBP and the TAF2 250. We will talk about this later in this presentation too. And the eraser protein for histone acetylation is histone deacetylase. So our central hypothesis when we start this project is to test that histone modifications coordinate the phenylalanine and VOC metabolite networks during flower development, hence to control the emission of the floral VOC. To test this hypothesis, the first thing we do is we profile the transcriptome and the global chromatin modification pattern during the petunia flower development. So we sampled at 2 stages. One is day 0 at bud stage when the flower is closed and is not making a lot of the VOCs at all. And second stage is day 2 of emphasis when the flower is fully open and the emission of the VOC rich is peak and is ready for pollination. So at these 2 stages, we harvest the corolla tissue only because that's where the emission of the scent happened. And we did histone ChIP-seq targeting at 2 different histone marks, H3K4 trimethylation and H3K9 acetylation. And from here, we identify hundreds to thousands of genes that has changing level of histone modification between these 2 developmental stages. So specifically, when we say hypermethylated, that means these 800 genes has increased level of H3K4 trimethylation on day 2 versus day 0. And when we say there is hyperacetylation, that means there is 4,000 genes with increased level of K9 acetylation on day 2 versus day 0. And to see how that may associate with gene expression, we also analyzed RNA-seq data harvested from the same developmental stage of plants grow under the same condition with RNA-seq. And from RNA-seq, we identify about 3,000 genes upregulated and 4,000 genes downregulated from day 0 to day 2. So first, we compare those different gene lists identified from the epigenome side, as well as identified from the transcriptome side. And what we saw first is there is a significant overlap of genes between the genes that are upregulated at mRNA level from day 0 to day 2, with the genes with increased K9 acetylation from day 0 to day 2. And similarly, there is a significant overlap between the induced genes and the genes with increased K4 trimethylation from day 0 to day 2. So those are not surprising. It's more like what we expected because the K4 trimethylation and K9 acetylation are both known to be associated with gene activation. But when we look specifically into what genes has increased K9 acetylation versus what genes has increased K4 trimethylation, things become a little bit more interesting. So here we're looking at the Galton analysis result, and we are looking at the significant enriched Galtons among the genes with increased K9 acetylation versus Galtons among genes with increased K4 trimethylation. And this is a Revigo plot. In this plot, each circle here is a significant Galton. And if the circle is big or the circle has a color towards green and blue, that means that Galton is really significant. And within this semantic space, the similar sounding Galtons are tightly clustered together. So a cluster of related or similar Galtons are only labeled with one representative term. That's a way of simplifying the Galton analysis result and for us to see what is going on. So as you may notice, there are some shared Galtons between these two groups, for example, transport and post-transcriptional regulation. But for the metabolite Galton specifically related to aromatic amino acid biosynthesis, this is only enriched among genes with increased K9 acetylation, but not with genes with increased K4 trimethylation. So the aromatic amino acid Galton is related to the synthesis of phenylalanine, which is a precursor for the VOC biosynthesis. So this is our first indication that K9 acetylation, but not K4 trimethylation, are specifically associated with the activation of the Phe and VOC metabolite network. But there is limit of Galton, and especially most of these Galton are derived from a homologue with Arabidopsis, where the study of secondary metabolism is rather limited. So the next step, we specifically look at the pathways that are directing the carbon flux towards the VOC formation. So at the beginning of the talk, I show you the primary and secondary metabolic pathway involved in the synthesis of Phe, phenylalanine, and the VOC. And this is, again, a simplified view of that. The primary metabolism network part is the shikimate and Phe biosynthesis pathway, which leads to the generation of phenylalanine, which is then converted to cinnamate and then 4-coumaryl-CoA. And both phenylalanine and cinnamate can be used to convert it to floral VOCs, benzenoid and phenylpropanoid VOCs through this pathway. And the 4-coumaryl-CoA can then enter the monolignyl pathway to generate monolignyl intermediates, which also can be used to make a eugenol and isoeugenol, which are also part of the floral VOC. So here, all the compounds that can function as a floral VOC are highlighted in red here. Therefore, these 4 pathways highlighted in various shades of green and yellowish, those are pathways that can direct carbon flux towards VOC formation. And in flower, there's also other metabolic pathways that actually compete with VOC formation and then direct the carbon flux away from VOC formation. One of these is the anthocyanin biosynthesis pathway. It can use the precursor for fumaryl-CoA to make anthocyanin. And the other is lignin biosynthesis pathway, the polymerization pathway. It competes with the eugenol and isoeugenol for the monolignyl intermediate to make lignin. So for each of these pathways here, we did a detailed analysis to see how that pathway is regulated at the transcript level as well as at the histone modification level. I will just show you the result from the shikimate and Phi biosynthesis pathway as an example. So this is the shikimate pathway that eventually leads to the generation of phenylalanine. And each of these enzymatic steps is categorized by enzymes that are encoded by multiple copies of genes. So first, we overlay the expression level of those genes onto this pathway, which is indicated by this little square here. A green color in this little square shows that those genes are upregulated from day 0 to day 2. While a magenta color means those genes are downregulated from day 0 to day 2. So we can see that quite obviously, every step there is at least one copy of genes, sometimes multiple copies of genes are induced from day 0 to day 2. Therefore, we see an enrichment of induced genes in this pathway. And then we overlay the histone modification data onto this pathway. And we can see that, again, for majority of the steps here, we can see at least one gene copy has increased level of K9 acetylation. So there is an enrichment of genes with increased K9 acetylation in this pathway. And by contrast, there is no enrichment of genes with increasing K4 trimethylation in this pathway. So this is the example to show that the shikimate pathway, which provide a precursor phenylalanine, are enriched with gene activation in the form of increased mRNA as well as increased K9 acetylation when the flower opens from day 0 to day 2. And this is the example of 2 genes, EPSPS1 here and DAHP1 here. Their expression level, the RNA-seq value, are increased from day 0 to day 2, and their K9 acetylation, the level from the ChIP-seq also increased from day 0 to day 2. So we did that analysis for all these 4 pathways here, and we see a pretty clear image, which is for all the pathways that directing the carbon flux towards the VOC emission. We see an enrichment of genes with increased expression level as well as increasing K9 acetylation. And the pathways that direct carbon flux away from VOC biosynthesis, we see a downregulation of gene expression. So this suggests that H3K9 acetylation might have a function to direct carbon flux towards VOC formation. And the next question we want to ask is, what is the point of regulation within each of these pathways? To answer that, we actually created a network view of this metabolic pathway. And so here, the metabolites are shown in the circle. And then the enzymes or the genes encoding the enzymes are shown as the square. So for example, these 3 copies of genes encoding the enzyme to convert this metabolite to this metabolite. And this is a combined networked image of the primary metabolism as well as secondary metabolism that are relevant to the Phi and VOC biosynthesis. So on this part, it is the primary metabolism, which is the shikimate and Phi biosynthesis, which leads to the generation of phenylalanine. And then phenylalanine, as I said, can be used as a precursor for the secondary metabolism, to generate a VOC, and which is the part of the metabolic network shaded in green here. So all the VOC compounds that can be emitted from the flower to contribute to the scent is shaded in orange here. So in the flower, in addition to the VOC biosynthesis pathway, there is also two other metabolic pathways that can compete with the VOC biosynthesis for the intermediate, which is the lignin biosynthesis and the anthocyanin biosynthesis, highlighted with the brown shade or the purple shade here. Because of those competing metabolic states, for a certain metabolite here, they serve as the divergent point to enter different metabolic fate. For example, cinnamate, it can be used to generate VOC, or it's the branching point to contribute a carbon towards the lignin biosynthesis, as well as anthocyanin biosynthesis. Another branching point like that is the 4-coumaryl-CoA that can be used for lignin biosynthesis, or it can be converted to anthocyanin. And if it enters the lignin biosynthesis pathway, it first leads to the generation of the monolignol intermediate, which are these three. And then one of the monolignol intermediate, coniferol alcohol, this again is a branching point because it can be used for the synthesis of lignin, but it can also be used to convert it to eugenol and isoeugenol, which is the part of the VOC biosynthesis pathway. So when we look at the whole network, we can see there is these 4 different divergent points highlighted in magenta here. Okay, so with this network structure, now we can overlay the information of chromatin regulation as well as mRNA induction onto this network, and we are highlighting the genes encoding enzymes. And those genes have increased K9 acetylation from day 0 to day 2, as well as increased mRNA level from day 0 and day 2. And those genes will be highlighted in green. So here is what it looks. So by looking at which point of this network is regulated at epigenetic level as well as transcript level, we can observe some different rules for points of chromatin regulation between the primary metabolism and the secondary metabolic network. For the primary metabolic network, we can see that almost every enzymatic step is regulated at chromatin level, 80% of the steps. But that's quite different for the secondary metabolic network, where the regulation point is more sparse. It's only regulated at specific enzymatic steps, about 50% of the enzymatic steps. And when we look which 50%, we notice that very often you observe the regulation occurs before and or after the divergent point, which actually makes sense because the enzyme that leads to the generation of the shared metabolites need to be upregulated to make more of the shared metabolites. And when the level of the shared metabolites increase, the enzyme that after that step also need to be increased to make sure the metabolites enter the fate that is useful for the flower, which is the VOC biosynthesis pathway. So far, we observed some association between K9 acetylation increasing with the gene activation during flower development. We also observed some interesting patterns of the choice of regulation, but that's only association and not causality. So next, we need to test whether histone acetylation is indeed required for activating the Phe and VOC gene networks. For that, we use chemical inhibitors for histone acetyltransferase, which are the writer proteins for histone acetylation. And as I said in the very beginning, there are 3 gene families of histone acetyltransferase, short as HAP, and GNAT and MIST can be inhibited by chemical inhibitor MB3, and CBP can be inhibited by C646. So we take an unopened flower and then feed it these chemical inhibitors. and then use qRT-PCR to monitor the gene expression level of quite a few genes in the Phi and VOC gene networks on day 0, day 1, and day 2. We can see that in the mock sample, which is the black line here, the expression level of those genes are low in day 0 and increase from day 0 and to day 2, which is what we expected. And when we treat flower with MB3, which is the inhibitor of GNAT and MIST, we can see that the gene activation is significantly reduced. But when we treat flower with C646, which is the inhibitor for CBP, the activation is not affected. So this leads us to have a conclusion that a GNAT and MIST family of HAT is perhaps involved in deposit H3K9 acetylation on those genes in this network, which enable the activation of VOC synthesis. Another question is, which hat is that? So we look at all the hats in the petunia genome and plot their expression level from day 0 to day 2. And we notice that ELP3 is quite unique because it has a huge induction compared to the rest of the gene family from day 0 to day 2. So in petunia flower, we use RNAi technology to transiently knock down the expression level of ELP3. So you can see here, when you look at the expression level, the expression level of ELP3 is reduced to about lower than 40% of the wild type level in the empty vector control. So after we knock down ELP3, we use qRT-PCR to assay the expression level of all the other genes that are involved in the VOC synthesis. And we can see that for those genes, their expression level is also downregulated significantly compared to the empty vector control. And more importantly, in the RNAi line where ELP3 is downregulated, we also measured a downregulated level of VOC emission compared to the anti-vector control. So these serve as the evidence to support that ELP3, or histone acetyltransferase, is required for the VOC network activation as well as for the VOC emission. So this is our final model that ELP3 and the H3K9 acetylation that it deposits perhaps involved in directing carbon flux towards VOC formation during flower development. And the networks that involved in competing with the VOC formation, those are repressed and they are lacking K9 acetylation modifications. So the take-home message that I hope I have convinced you with is that H3K9 acetylation deposited by ELP3 co-activates primary metabolite network and secondary metabolite network to generate floral volatiles during flower development. And what I found really interesting is there seems to be a different philosophy about the H3K9 acetylation on the primary metabolism and secondary metabolism. The primary metabolism seems to have a more prevalent modification of K9 acetylation, and the secondary metabolism seems to have a more specific target choice, and those target choices seem to happen at nodes that are critical for directing the carbon flow towards the VOC formation, usually before and after the divergent point of the metabolic network. So with that, I'd like to acknowledge the people contributing to this work. And this is a collaborative work from my lab and then Natalia's lab. And this is a view of my lab from the horticulture garden at Purdue. And this is our 100-year-old horticulture building, and my lab is here. So we have a very good view of the garden. And Ryan Patrick is the leading author contributing to this manuscript, and he lead the effort of this research. And this is what our group meeting looks like in this pandemic era in the last 1 year. And then this work is not possible without the contribution with collaborator Natalia Gidereva and her postdoc David Huang. And fundings are from USDA, NSF, and the Purdue Seed Grant. So with that, I will stop in this summary slide and take any questions.
So thank you, Ying, for a really nice talk. So I'd like to invite questions from the floor. So you have two options. If you're happy to unmute and just go ahead and ask your questions direct to Ying, then please feel free to do so. If not, you can type your questions into the chat box, and I'm happy to ask those questions on your behalf. So let me just see if we've... Okay, so perhaps while waiting for some questions to come through, maybe I can start if you don't mind, you. So we know that volatiles are actually important for quite a lot of plant-insect interactions beyond pollination. You know, they're also important in attracting predators, etc. If plants are being attacked by pests. So do you have any evidence, or have you looked at whether other volatile release is controlled via a similar mechanism, or indeed whether other volatile pathways are controlled via similar mechanisms?
I agree with what you said. I haven't ventured into other VOC yet to see whether similar mechanism is there, but I wouldn't be surprised. So another important VOC is, for example, the fruit VOC that contributing to the smell and even the taste. And although people hasn't specifically looked into histone modification, but there is a paper showing that DNA methylation, another form of epigenetic regulation, involved in control of VOC during fruit storage, and that is done in tomato, I think. So I would love to look into whether similar mechanisms are also functioned in control the VOC in other systems, or even, you know, the secondary metabolic pathways in general. My guess is it may not be exactly the same histone modification or epigenetic regulator, but it wouldn't be surprising to me that regulatory mechanism function at the chromatin level contribute to the turning on and turning off of those metabolic pathways when it's needed, because this is a mechanism make a lot of sense to coordinate multiple enzymes at a specific time and tissue required.
Okay, thanks. So again, I'll just offer an opportunity for questions from the floor. So nothing in the chat box just now, and I can't see whether anyone's wishing to ask anything to the floor. So, so if you don't mind, perhaps I will go again. Then.
Sure, go ahead.
So I mean, obviously, you know, there are other levels of regulation which might occur. One level being transcription factors, obviously, and are you seeing any evidence that the transcription factors themselves are also subject to this epigenetic regulation?
Yeah.
And how do those two levels of regulation integrate? Do you have any feelings about that?
Yeah. Very good question. I think my apology of not mentioning any transcription factor when I give the background introduction. Actually, it is quite well known that there is a few meat transcription factors that are involved in activating the genes in the VOC biosynthesis. So when you talk about the histone modification, I see the transcription regulation and the epigenetic regulation as two mechanisms go hand in hand, because many times the epigenetic regulator, the protein itself, does not really have ability of identifying specific DNA motif in the genome, right? So when you ask the question, how does this ELP3 know to go to this gene loci to activate the gene? My guess, and it's also other similar research have shown that it's a transcription factor, find the target genes through recognizing the cis regulatory motif, and then recruit the epigenetic machinery to deal. But it also can go the other way, that the epigenetic modification occurs, created a favorable local chromatin environment for the transcription factor to bind. So these two mechanisms really work very close together. And going forward, we are kind of interested to do a more detailed analysis of, for example, how ELP3 target and recognize the genes in the genome. And we expect to probably get a hint of some transcription factor working with it. For example, when you analyze the promoter of the ELP3 target, you probably will see an enrichment of cis regulatory motif, and quite possible is the meat binding motif, because the non-regulators or non-transcription factor regulating this is meat family. So that's how I see these two mechanisms. They're really integrated.
So then, I mean, what else does the ELP, or what other genes do you think are under regulation via ELP3? I mean, obviously, clearly you focus very much on these genes or proteins involved in the volatile biosynthetic pathway. But in your ELP knockdowns, are there other pathways that you're observing, or any other specific enrichment that you're observing, or lack of enrichment, as it were?
We haven't done a specific ELP targeted genomics research. If you notice, we presented here, it's just day 0 to day 2, right? And it will be like to make our ELP knockdown line, or to do a cheat towards ELP to see what it binds, and then you will see what ELP regulates beyond this pathway. Right now, as far as ELP goes, we just got a hint that this is the guy and affecting this pathway. Yeah, the next question is what else does it do? And I think that's the next project.
Okay, so we are approaching our time. I'll just give the audience one last opportunity to ask any questions. And I don't think anyone's brave enough to go on camera, Ying. And you must have given a fantastic talk that answered everyone's questions.
Yeah,
so with that, I'd just like to take this opportunity to thank you for giving us this seminar, and to thank the audience for joining. So thank you very much.
Thank you, Rob, and thanks everyone.
Thank you.